All right, welcome to the section 2 notes from chapter 7. We're going to start out with this first objective, describe different types of allele interactions. The interaction that we're going to do in this video is going to be incomplete dominance. Incomplete dominance is going to be an example that's relevant to snapdragons and their flower colors, and also betta fish, possibly as well. Last time around when we had flowers, we had homozygous dominant, which led to that dominant red trait. We had homozygous recessive, leading to that recessive trait white. But when we had that heterozygous individual, the dominant trait always showed through, right? It was red. In this case, in incomplete dominance, that dominant trait and the recessive trait mix. And so you're going to end up with some intermediate color. In this case, it's going to be pink. So dominant's red, recessive is white, in between is going to be pink. So all three possible genotypes lead to three different possible phenotypes. So if I try doing a cross between two heterozygous flowers, both of which would be pink, I'm going to get results when it's all said and done, and I need to be able to predict outcomes. So I fill in my Punnett square. I again get these different options, same pattern as before, but instead of having a 3-to-1 ratio, red to white, I'm going to have a 1 to 2 to 3 ratio, because this top left offspring is going to be red, bottom right is going to be white, and the other two heterozygous individuals are going to be that pink color that's in between. So we have probabilities for those three different colors out of this. 1 out of 4 are going to be red, 2 out of 4, or 1/2 of the offspring are going to be pink, and 1/4 of the offspring are going to be white. So the big difference here, this incomplete dominance, there is an intermediate phenotype that shows up when you're that heterozygous genotype.